So for the BNS Chapter 3 Segment 2 outline, we're starting on ionic mechanisms underlying the action potentials. So the first thing to note is that the English neuroscientists Alan Hodgkin and Andrew Huxley use squid axons to establish that axon potentials are created by the movement of sodium into the cell through channels that open in the membrane. These channels switch from potassium-dependent to sodium-dependent states, then back to the resting state. And this occurs through voltage-gated ion channels. How these action potentials are mediated by voltage-gated sodium channels starts with the opening of potassium channels that create a resting potential. This then has any depolarizing forces that will bring the membrane closer to the threshold. At threshold, voltage-gated sodium channels open, which will cause a rapid change in polarity, which can mediate action potentials. These sodium channels will close automatically, and these gated potassium channels then open, which will repolarize the cell. All gated channels close, and then the cell will finally return to its normal resting potential. A voltage-gated sodium channel is just a channel that opens or closes in response to changes in voltage of the local membrane potential, and it's what mediates action potentials. It's generally closed unless it reaches threshold, and when threshold is reached, only a few channels open, letting in sodium. As the ions enter, the depolarization continues, which will open more channels. Each of these channels are open for about only a millisecond. The resting potential is restored when the potassium re-enters the cell, when the channels reopen. These channels have refractory periods, which are inactive or exhausted axonal membranes where these cells are completely unresponsive. There's two phases to refractoriness. The first is absolute refractory, which is a brief period of complete insensitivity to stimuli where the sodium channels are completely unresponsive. There's relative refractory phase as well, which is a period of reduced sensitivity, during which only strong stimulation can produce an action potential. The flow of the potassium ions has the temporary hyperpolarization of the neuron, so a stronger stimulus is needed to reach that threshold value. The transmission of these axon potentials are generally limited to the axons, and these potentials start in the axon hillock, where it regenerates itself down the length of the axon. So we're having a little break talking about box 3.1, which is voltage clamping and patch clamping. Voltage clamping is the use of electrodes to inject current into the axon or neuron to keep the membrane potential at a set value. It uses an apparatus that measures how much has to be injected to counteract any ion channels opening. The apparatus consists of two electrodes, where one is in the axon and the other is in the bath surrounding the axon. So the first step of that would be that the axon electrode measures the membrane potential and is connected to the voltage clamp amplifier. The voltage clamp amplifier compares membrane potential to the desired command potential, and when the membrane potential is different from the command potential, the clamp amplifier injects current through the axon of the second electrode, causing the membrane potential to become the same as the command potential. This current then flows back into the axon and thus across the membrane, allowing it to be measured. Patch clamping is a type of voltage clamping, which is used to monitor the flow across a tiny patch of membrane taken from the neuron. This gives some more detailed information and can detect how many channels are open and what specific ion channels are working. There's three types of patch clamping: cell-attached recordings, whole cell recordings, and outside-out recording patches. So moving back to action potentials and their propagation through the axon, or through the axons. The axon potential regenerates along the length of an axon, so it depolarizes the next adjacent axon segment, and that continues down to the axon terminals. The axon hillock sends that information to the axon terminals. The axon hillock can't backfire because there's a small number of sodium channels on it, so it can't conduct that velocity backwards. Conduction velocity describes the speed of which an action potential is propagated along the length of an axon. It varies with the diameter of the cell, so a larger axon allows depolarizations to spread faster through the interior due to the ions having more space to flow. These can be as fast as 120 meters per second. Myelin sheathing also affects the speed of conduction, making it quicker. Myelin interrupted by nodes of Ranvier, which the action potentials will jump over. There's also saltatory conduction, which is a form of conduction that is characteristic of myelinated axons, in which the action potential jumps from one node of Ranvier to the next. An unmyelinated axon would need to be 100 times larger in volume than a myelinated axon to have the same speed of conduction. We now move to box 3.2, which is changing the channel. Channelopathy is a genetic abnormality of ion channels, which causes a variety of symptoms, including mutations of genes that encode certain channels, and this can cause things like epilepsy. Tetrodotoxin is a toxin from pufferfish ovaries that block the voltage-gated sodium channels, which prevent action potential conduction. This can cause paralysis and death. There's also saxitoxin, which is an animal toxin that blocks sodium channels when applied to the outer surface of the cell. And these come from red tide bloom of allergies. The last toxin we're talking about is batroxytoxin, which is secreted by poison arrow frogs, and it selectively interferes with sodium channels. We also do use drugs that purposely block sodium channels like lidocaine, which will selectively block the channels in the area which it's injected. So moving back to synapses, we move to the next section, which is synapses cause graded local changes in the postsynaptic membrane potential. So as we said before, action potentials are propagated down the axon, and it goes down to where the axon splits into branches, and the axon potential will be recreated in each of those branches until it reaches the different axon terminals of each branch. These axon potentials are then converted into chemical signals as the axon terminal releases a neurotransmitter. So synapses can also be excitatory or inhibitory. So neurotransmitters released into the synapse briefly alter the membrane potential of postsynaptic cells, causing postsynaptic potentials. Postsynaptic potential is a local potential that is initiated by stimulation at a synapse, and these can vary in amplitude and will spread passively across the cell membrane, meaning that it'll decrease in strength with time and distance. You can study and compare the effects of excitatory versus inhibitory synapses on the local membrane potentials of postsynaptic cells. Excitatory synapses will have normal action potentials and will cause depolarization in the postsynaptic cell, while inhibitory have normal action potentials as well, but will cause hyperpolarization in postsynaptic neurons. To stimulate the presynaptic neuron will cause it to produce an all-or-nothing action potential that will spread to the end of the axon, which will then release that neurotransmitter. After that brief delay that we talked about, the postsynaptic cells have a small depolarization, which will open the channels to let the sodium ions in. Excitatory postsynaptic potentials are depolarizing potentials in the postsynaptic neuron that are caused by excitatory connections. EPSPs increase the probability that the postsynaptic neuron will fire an action potential. You can also combine the effect of excitatory synapses needed to elicit an action potential in a postsynaptic cell. If enough of this occurs, it can combine to produce a depolarization large enough to reach threshold and trigger an action potential. Postsynaptic depolarizations usually begin within a half a millisecond after an action potential arrives at a presynaptic terminal. So there's a synaptic delay, which is the brief delay between the arrival of an action potential in the axon terminal, at the axon terminal, and the creation of a postsynaptic potential. This time just accounts for the needed time for the neurotransmitters to be released and diffuse across the synapse. All neurons use the same kind of propagated signals, but the effects on the postsynaptic cells can be very different. With inhibitory synapses, the firing causes an increase in resting membrane potential, which is hyperpolarization, and it moves away from threshold and decreases the probability that a neuron will fire an action potential. An inhibitory postsynaptic potential, by definition, is a hyperpolarizing potential in the postsynaptic neuron that is caused by inhibitory connections. And as I said before, IPSPs will decrease the probability that postsynaptic neurons will fire an action potential. These are usually caused by the opening of channels that let chlorine ions into the cell, making the inside of the cell more negative. Inhibitory postsynaptic potentials aid in neural processing of lots of different information. So with overexcitation of neurons, that can cause seizures, where underexcitation can cause coma and death. So there's a nice sweet spot where you need to have inhibitory and excitatory synapses. So now we're getting into spatial summation and temporal summation, which integrate synaptic inputs. So neurons integrate and transfer messages received that play a role in information processing. As the book describes it, it's the use of neural algebra, where each neuron is going to add or subtract inputs it receives from other neurons. So postsynaptic potentials are usually caused by transmitter chemicals that can be either depolarizing or hyperpolarizing. So there's graded EPSPs and IPSPs that spread passively over neurons. And whether an action potential is fired is determined whether a depolarization exceeds threshold just beyond the axon hillock, and it will allow for that action potential to begin. Going into the actual summations, excitatory and inhibitory neurons are able to cancel each other out, but more excitatory inputs at once can overcome the inhibition. That means that sometimes excitatory signals will pair up, which is what is required to reach threshold and fire an action potential. With this said, distance does matter. Two EPSPs closer to the axon hillock have a greater summation than the two EPSPs farther away from the axon hillock. This is known as spatial summation, which is by definition the summation of at the axon hillock of postsynaptic potentials from across the cell body. If the summation reaches threshold, an action potential will be triggered. There's another type of summation as well, which is temporal summation, which is the summation of postsynaptic potentials that reach the axon hillock at different times. But the closer in time the potentials occur, the more complete the summations. So what that means is that if you have two action potentials that happen in a closer amount of time, there may be a decent amount of overlap that will allow the synapse to reach threshold. The farther out on a dendrite a potential is produced, the less effective it's going to be, the potential should have on the axon hillock, because the potential decreases in amplitude as it passively spreads. In some neurons that have synapses that are farther away from the axon hillock, it'll compensate by producing larger postsynaptic potentials. Some of them have some dendritic integration zones, which have voltage-gated ion channels, which can sum and amplify local postsynaptic potentials, including the impact on the axon hillock. Another thing is that they'll have glial cells, which can help strengthen the postsynaptic potentials by preventing neurotransmitters from leaking out of the cell. So both of those can help with synapses that are farther away from the axon hillock and need some extra help to form some action potentials.